up till now, if you look at it, uh, maybe of course the lithium ion battery was one example of a product. But if you look at the Haber process, asymmetric hybridization, they are all processes. Okay, we've covered a lot of processes, and or a quest for understanding Ertl and Langmuir and people like that. Of course, Langmuir also came up with a better product through his understanding. But now we will go to how a drug is actually conceived. Okay, and this, of course, you can imagine. Once you have made a drug, then of course it can be made by many different processes, and we have been quite good at making these drugs through efficient processes. Ask the Chinese. But many times, most times, I find that we have not really conceived the product. So let me tell you how a drug was conceived, and nothing more important than a hard drug. So I've selected a hard drug, and the other reason for selecting was that, as I told you, I have either been trying to teach you from the Nobel lectures, or from patents, or from people or whom I may be in touch with, and so I can get a first-hand idea of what was in their mind. And here, of course, the first part of it is by Sir James Black, who won the Nobel Prize for in 1988 for the first rational design of a hard drug. And later on, we will come to another very important drug, which for me it was good fortune that it was discovered by my roommate in Princeton, John Dancia. And so I will tell you about that, because I've been able to get all the information, what was really going on in the mind. Okay, and so both will be covered. So, and James Black, you can see he was knighted for this work, Sir James Black, and he's one of those rare people, and you can see what it says. I just took this from Wikipedia, and is considered by many to be one of the most important contributions to clinical medicine and pharmacology of the 20th century. You're not talking about a decade, of the century, you know. That is the kind of impact this drug had. It's very rare for a person to go and discover another drug, you know, and he's one of those. He also discovered this molecule called cimetidine, which was very popular for stomach ulcers. So James Black was extraordinary. He did both, and you hear about this AstraZeneca jab. So that Zeneca was earlier basically a part of ICI, and it was spun off as a separate company called Zeneca in 1993. And so they had a great tradition of drug discovery. Okay. And of course, later on, vaccine discovery with Oxford, which they did last year. Okay, now let us try and understand a few things. Now, when we are talking about covering something on drug discovery, and neither am I a guy who's got expertise in life science, nor do you probably. So sometimes, you know, the jargon is difficult. Okay. But I found that you can always go and look up some of these terms, and you will be able to figure out what really they're talking about. Okay. Now, so he begins his Nobel lecture. He's talking about clinically angina pectoris. And then I said, what is angina pectoris? It is basically a temporary chest discomfort that occurs when the heart is not getting enough blood. That's what it is. Okay. That's called angina pain. All right. And it was known to be precipitated by anxiety. So, if you are very anxious, those kinds of things can sometimes cause a surge in adrenaline. And it was known that those kinds of things can give you this pain. And another is, of course, if you are going through an emotional upheaval, those kinds of things can also cause this. Or if you exercise a lot, you know, this very very rigorous exercise tends to the body tends to generate a lot of adrenaline. And so what was also known was that if there was a chest pain and you're not very sure, the doctor was not sure as to why you're getting your chest pain, that they would inject some adrenaline, right? And see if your chest pain is actually getting aggravated. And that was then being used as a diagnostic test to know whether what exactly is the cause of your chest pain. Now, another thing which was known was that adrenaline is produced by the thyroid gland. And so, if there was a very severe chest pain, what people would do was remove some part of the thyroid gland. Okay, so that's called partial thyroidectomy. Okay, and that helped to relieve, some of this angina pectoris. That was also known. Now, Black also found that the fast beating of the heart, okay, normally a heart should beat between 60 to 100 beats per minute, but sometimes the heart starts racing. And what he found is that, or he surmised, he said that could it be that this very fast beating of the heart may be also connecting these disorders. So again, you can see it's a hypothesis. At that time, he didn't know much more about it, but you begin somewhere, and he began with a hypothesis. Now, let me also tell you that it's not as if there was no heart drug which was known. This is another remarkable thing which you would have seen literally in everything. Whatever was done by people, somebody sometime before had done something. It's extraordinary, everywhere, and that's why it's called research. You're really researching where things have already been done, and those guys who really did it for the first time are absolute genius. And but many times, there was an accidental discovery. So what was very well known was nitroglycerin. This is nitroglycerin. And where do you use nitroglycerin very commonly? Can one of you say? It's very common. Large volume application.
It is an explosive.
Explosive. Absolutely. Nitroglycerin. Okay, dynamite. That's what Nobel got his Nobel Prize for nitroglycerin. Okay, and making it safer. Okay, and that same nitroglycerin, which is so famous as a explosive, people found that actually it relieves this angina pain. Okay, and later on, these people, William Murrell, you can see 1879. We are talking about 140 years ago. Okay, basically was able to show that these nitrated compounds like nitroglycerin tend to break down and generate N-oxide. Okay, and it was the N-oxide which was really doing the job of relieving this pain. Another very interesting thing, when nitroglycerin is being sold for medical application, they don't call it nitroglycerin. They call it glyceryl trinitrate. Partly, I think, because if you say nitroglycerin, people will get scared. Oh my God, they're giving me explosives to eat, and so you just conveniently change the name. And so anyway, this was known. But what was also known was that this nitroglycerin has side effects, and it can give you a terrible headache. Okay, and you can get a facial flush. I know that because at one time, as I told you, I was doing work on explosives, and so I used to occasionally go to the nitroglycerin plant. They were making dynamite, and if I came out of that plant, I couldn't stand it. I mean, you felt so horrible with a headache, and it would take me a good few hours to get rid of my headache. So there is a bad side effect of nitroglycerin, and what people knew was that what possibly happens is that some of these arteries, etc., get clogged or restricted, and these molecules, like N oxide, etc., tend to dilate the arteries, the coronary arteries, and so it's a supply and demand thing. So you are basically the heart wants more blood, it's racing fast, and then you're trying to push more blood into the heart. Okay, you have to meet the demand, so you're trying to fulfill the demand for blood. Actually, not for blood, actually for oxygen. That's what you're trying to fulfill the demand for. Now, what was also known was this particular compound. In fact, this compound is even today popular, dipyridamole, and this was again something which was known to have a positive effect, and it had the advantage that it did not give you this headache and stuff like that. So something was known, but not a very successful drug. There was some positive effect, but it really could not be called as a cure. You know. Okay. Now there were other people who did very clever work. For example, these people in 1958, and that was the time when James Black started his work around 57, 58. What they did was they said, what blood? It's all about finally oxygen. You know, okay. A body needs more oxygen, and the heart is pumping so to provide that oxygen to the body, to the cells. And so what these guys did was that they supplied hyperbaric oxygen, oxygen at 2 atmosphere pressure, and they found that this very rapid beating of the heart, etc., actually they could control by giving oxygen which was more concentrated with a higher partial pressure. Okay, so that was also known. Now, then, what Black said is something quite revolutionary. Till that time, everybody was basically trying to fulfill the demand. I need more blood, dilate the vessels. I need more oxygen, pump hyperbaric oxygen. Okay. But what Black said is, hey, why are we so hung up on trying to always fulfill the demand? Why don't we just slow down the heart? And he went in what I would call, and we'll be discussing when we talk about the TRIZ methodology, the other way around. He went the other way. All of these guys were trying to supply more, and he said, "Let me reduce the demand." And of course, if you try to reduce demand too much, then you may die also. Okay, because that's what happens. Now, when you're dead, the heart doesn't beat at all. Okay, so you're dead. Obviously, he wasn't talking about going to those levels of slowing down. He just wanted to slow it down, and especially for people whose heart was racing. Okay, and he says, might not an equivalently small decrease in the myocardial, and myocardial means related to heart muscles, demand for oxygen be just as effective? That was my question. Myocardial oxygen consumption is determined by the work of the heart and is a function of arterial blood pressure and heart rate. Lowering blood pressure by systemic vasodilation, when I dilate the arteries, might dangerously reduce the perfusion pressure and blood flow through diseased narrowed coronary arteries. He said, you're trying to do this and there's a diseased artery. You might be creating more havoc in trying to dilate those arteries. And he said that be careful. And then he said, indeed, hypotension was known to be able to induce a heart attack. Hypotension will happen when I have actually dilated the arteries a lot, and so my blood pressure has come down. But it was known that that also can induce a heart attack. So his reasoning was, can I go the other way around? We will just discuss how a new concept was born, okay, and that was the concept of receptors. Uh, these people, Langley, okay, Ehrlich and Langley, between 1900 and 1948, published some seminal papers, and they were able to show, that there are certain receptors in our organs, and those are the receptors on which sometimes good things or bad things can go and sit, and that then leads to a good effect or a bad effect on the organ. And so they had basically worked it out, and later on in 1948, another person by the name of Alquist, he was able to also show that there isn't only one type of receptor in the body, that there are, and he was able to identify at least two types, which he then called as alpha and beta, two distinct receptors, which are present in the body. So 1948, so a good 10 years before James Black actually began his work. So James Black was already aware of this work on receptors. The other was, and we will discuss, that there was a particular compound which was shown to have a good effect on this blood pressure and all, and it was believed to be acting through some kind of a binding to receptors. Okay, so these knowledge were already known to James Black. Okay, so let us quickly try and understand what is a receptor. So, like, let's say if this is a cardiac cell and this is a receptor, and let's say in the cardiac cell, what they found is that actually you have the beta receptors, not the alpha. And effectively, what happens is, like, for example, adrenaline, okay, which was causing this problem of angina pectoris. What they found is adrenaline, etc., can go and sit on these receptors, and that's what actually causes all these problems. Okay, so what you then try to do is that you develop a strategy to dislodge the problematic molecule from the receptor by introducing another molecule which goes and binds more strongly to the receptor than the problematic molecule. Okay, and this molecule otherwise is a benign molecule which does not have any adverse effect. All right, so that then, for example, if this was a beta receptor and I was concerned about adrenaline going and sitting on it, then I would try to block the entry of adrenaline, and the blocker is basically what is known as a beta blocker. Okay, that's what it is. So effectively now the adrenaline cannot go onto this, and it's out. Okay? And you have more or less solved your problem. Now let us also look at the classes of molecules, and we saw when we covered Noel's work, we saw L-DOPA. You remember this L-DOPA? Okay, it's what? It's an amino acid. It's an alpha amino acid. Okay, and you have got this catechol. Is basically a catechol derivative. Okay, now catechol actually produces many different kinds of molecules in the body. So you can have catechol amine. It's a primary amine, all right, with a catechol. This is your adrenaline. Again, you can say it's a catechol derivative. It's also an amine derivative, except the difference between catecholamine and this is that this is a secondary amine and there is a hydroxylation at this carbon. So that is adrenaline. There is also something known as noradrenaline, which is it's a primary amine. It's like catecholamine. It's hydroxylated like the adrenaline. Okay, so you can see that these are all closely related classes of molecules. And then this compound was also made synthetically, which is known as isoprenaline, and it was being used for the treatment of bradycardia, slowing the heart rate. But the problem was it was also being used for treatment of asthma, and it was non-selective beta receptor, means it could also go and attack the alpha receptors. And if it does that, then it can solve one problem but create another. And so many times, when you are looking at and talking about side effects in drugs, it is because of their non-selective binding to a receptor, and it goes and non-selectively goes and binds to some other receptor where it should not have gone and sat. So isoprenaline was that kind of a molecule. At that time, another very, very important thing was revealed around the time that James Black was working, that when we say uh, beta receptor is not one type of receptor. And they could subdivide the beta receptor into three different types of beta receptors: beta 1, beta 2, and beta 3. And it was known that the beta 1 receptor is the one which effectively works on adrenaline. And it was also known that these beta 1 receptors are located mainly in the heart and the kidneys. That was known. The beta 2 receptors were located mostly in the lungs, gastrointestinal tract, liver, uterus, you can see. And the beta 3 receptors were located in fat cells. So if you felt very thrilled that you have made a beta receptor, you need not be very thrilled, because it could have gone and sat on beta 2 and beta 3 as well. Okay. And so what would be our choice? Which receptor do we want?
Beta 1, right?
Beta 1. Okay, correct. So Black knew then that he needs to effectively make a blocker for beta 1 receptors. Okay, and a blocker is also known as an antagonist. It antagonizes the problematic molecule. I can also get an agonist. An agonist is one which almost like helps the problematic molecule to work even better. Means I'll actually aggravate the problem. Then it is known as an agonist. If it is contrary to the working of the problematic molecule like adrenaline, then I will call it an antagonist. So he says, so I started at ICI with a clear goal. I wanted to find a beta receptor antagonist. At that time, he said, let me first find a beta receptor. I'll see about beta 1, beta 2, beta 3 later on. He just wanted to begin with a beta receptor. I expected this to reduce pulse rates at rest and during exercise, and hoped that it would decrease the susceptibility of patients to angina pectoris. That was a hope. The unknown factor for me at that time was the significance of adrenaline's annoxiating activity. So, what happens is that there was something else. Adrenaline was also known to actually deprive the blood vessels of oxygen. So, I mean, let us not go into so much of technical stuff, but there were certain other things about adrenaline which he was not aware of at that time. And but what he knew was that if he can control this, that he might be able to slow down the heart, which is what he was targeting. Okay. And the other thing was that, like we saw Haber with his high pressure reactor, Langmuir with his McLeod gauge, and so many others, Sutin with his stop flow equipment, etc., Ertl with his surface spectroscopy, that similarly James Black was able to, he was the first who developed an equipment which could record both blood pressure and heart rate, because both were important. And at that time, no such instrument existed. Today, of course, it's all routine. You can routinely measure blood pressure and heart rate. But at that time, there was nothing. So he actually made that instrument. And so I think the reason why he was successful, whereas many of the others weren't, was that he had this much deeper knowledge on the functioning of potential leads, that he could measure both the blood pressure and the heart rate. Okay. And he put that to great use. As we said, this was already known, this DCI. Okay. But what James Black says is that these people who had discovered DCI, this is DCI. DCI is very interesting. Instead of catechol with the two hydroxyl groups, now you've got two chloride groups, two Cl groups. The rest of it, as you can see, is quite close to this structure. Okay, so you have got and this structure. So it was effectively this structure, except that OH and OH was replaced with Cl and Cl. Okay, they had made that compound. These people, Moran and Perkins. And Moran and Perkins reported that there was muscular contraction. Presumably, it had slowed down the heart. But what Black says is that no, Moran and Perkins are claiming things, but actually they have not really done what they ought to do. And so his studies seem to suggest that what Moran and Perkins had claimed was not correct. And what he says is, with a specialized testing protocol, Black found that DCI was as powerful a stimulant as isoprenaline of both the rate and force of beating of guinea pig heart. Maybe guinea pig heart was different from rat heart or mouse heart on which Moran and Perkins might have done their work. I don't know. And then he goes on to say that DCI was not the lead they were looking for. Okay. He says, yeah, I mean, they've done this, but this is not exactly what will solve our problem. Much as he seems to be criticizing this molecule, I find that he actually latched onto this molecule. And he just played around this molecule. And he says that over here, I shall never forget John Stephenson's reaction to this discovery. What was the discovery? The discovery of Moran and Perkins. That this stuff is possibly something that is affecting muscular contractions. If it was not a lead, what was he so excited about, he and his team? And then he says Stephenson had realized immediately that while a fused benzene ring would have similar steric and electronic properties to the two chlorine atoms, there was also the possible advantage of extended pi bonding. In other words, what they were trying to do, Stephenson and Black, was they wanted to make a derivative where the chlorides will not be there, and they felt that instead of that, if they introduce this phenyl ring, the phenyl ring will have the inductive effect of the Cls, okay, and the Cl, chloride is quite a big atom, okay, the Cl, the total amount of space that these will occupy in the 1-2 position was more or less similar to what would be occupied by this benzene ring. Okay, and so what they did was effectively converted from here to here. Okay, and that was the compound ICI 38174, which they nicknamed as nethalide for a time, but finally christened as pronethalol. And pronethalol was, and he says, was conceived in excitement and thrilled us at its birth. And then he says pronethalol was an antagonist. In other words, he maintains his claim that it was not something which Moran and Perkins had claimed. Okay, pronethalol was an antagonist without any sign of agonist activity in both atrial and ventricular heart chamber. Maybe what he found is that both chambers, atrial and ventricular, there was this kind of a activity of antagonistic activity of adrenaline. Okay, and so this is what he was wanting to do. He wanted to slow down the heart, and this particular molecule, which was conceived from this molecule, which apparently did not work, was successful, and that is what was known as pronethalol. But pronethalol had a very short life, and I'll come to that. Now, what I also want you to do is that when I'm going through these lectures, uh, what I find is that, uh, and partly it is the way that these people used to do discovery research. They used to make molecules. Okay, there was a group whose job was to keep making molecules. And then there was another group who would try out the molecule for all kinds of things, not necessarily for what you thought you wanted to make, but for many others. And in fact, for example, the birth of polythene, okay, was also related to something like this. They were trying out things for different applications. And now, what is very interesting is that you see the same moiety, very similar, quite similar to this, okay, not exactly identical, but it's also got a dichloro group, okay, and this molecule is what is used by the paint industry, okay, and effectively to for antifungal paints. Today you would have noticed that the paints, the coatings don't easily blacken. Earlier days, after every monsoon, the walls would look terribly black, and that's because a lot of fungus, etc., would grow on those paints. And Bayer were the people who actually introduced this particular molecule diuron, okay, and later on, ICI were able to, because ICI were very big in paints, Dulux paint, and so they were able to show, that diuron works extremely well as an antifungal agent, and they put it into paints, and they could keep the walls absolutely beautifully, almost like new. No fungus grew on it. And this is what is used by Asian Paints. This that name any company, they use it. Okay. The problem is that diuron today is having a lot of challenges. People feel that it's a toxic substance. And so the paint companies have been asked to look for some other molecule. They have been told that, look, I mean, we are not going to allow you to use diuron forever. And so the paint companies are scrambling to find an alternative. And I mean, my mind was going back and forth because I also happen to be a member of the Technology Council of Asian Paints. So I'm aware of all of this. And so I was just thinking if we can introduce a benzene ring instead of this, just like Stephenson had done, who knows, we might get the antifungal effect and we may not get the toxic effect of diuron. It's possible. I mean, it's all a wild speculation on my part, but I just thought that I'll tell you how people come up with ideas for drug discovery. Okay, they were jumping when pronethalol was made, but they soon found out that pronethalol also has problems. And he says over here, After pronethaolol, basal heart rate and force were reduced and the effects of adrenaline were abolished. So he had done a great job. However, the vasodilator effects of adrenaline were also blocked. Adrenaline had a good function that it could dilate the blood vessels and therefore it could allow blood also to flow into the heart. But if I now prevent that action of adrenaline and if it starts constricting the heart, then I have really not done something so great. I've slowed down the heart. On the other hand, I'm not making blood available to the heart. The body is not going to have enough blood and oxygen. And so he says, however, the vasodilator effects of adrenaline were also blocked, thus exposing the heart to a vasoconstrictor load mediated by the unblocked alpha receptors. The alpha receptors were in those arteries. And effectively, this pronethaolol they found is not a beta receptor alone. It was going and sitting on the beta receptor, but it was also going and sitting on alpha receptor and creating undesired effects. And then he says, pronethaolol always seemed to us to be a prototype drug, good enough to answer questions of principle, but not good enough to be marketable. So a large chemical group directed by Crowther was assembled to try to find a more active, safer replacement for pronethaolol. Pronethaolol definitely reduced the heart rate. It abolished the effect of adrenaline on the heart, but did not solve the problem of the vasoconstriction which they encountered. And remember that James Black was only a researcher, and this person, Crowther, was the research manager. So he was the one who was controlling the show. He was giving directions. And ideas were many times very, very intuitive. And what did they say? Look at what they say. And so when pronethaolol was not working and Crowther assembled a team, what they did was, this is another thing that you will learn also from Noyeri's lecture, that don't go and make radical changes. Okay? Make small changes. What was the small change that they made? In this molecule, this carbon was connected directly to this carbon. Okay? You had a CC bond. What they did was, they felt that this molecule is too hydrophobic. And maybe it has to be made a bit more hydrophilic. Okay? So they introduced a ether linkage, an oxygen here. And they just made this. Now, look at the dramatic effect that had the discovery of ICI 45520 propranolol. This is what propranolol is. A naphthyloxypropanolamine derivative was the result. That's what Crowther had asked them to do. They made all molecules, and this molecule turned out to be a super drug. It was far better than pronethaolol, but he says even this had some side effects. Okay, so it was definitely better than pronethaolol, abolished many of the side effects, but still had some side effects, and that was because it continued to be a non-selective beta blocker. What did they do after that? They said that they were suspecting that this compound, the side effects that were there, was because it was still very hydrophobic, because of this naphthyl group. And because of that, they said that it was probably inducing side effects. And so they wanted to basically make the molecule still more hydrophilic. Okay? And he says it was suspected that propranolol's centrally induced side effect could be due to its high lipophilicity. And what did they do? They then knocked off this extra phenyl ring which they had put in. Remember, almost going back to DCI. And what they did instead, was that they introduced an acyl amino group. So they made an NH2 over here and they acylated it, CH3CO, okay? And this molecule, of course, is quite hydrophilic, okay? And this was the first selective beta 1 receptor antagonist. It had very high selectivity for beta 1. Okay, and then he says para-acyl amino group was selected as the hydrophilic moiety, and this gave birth to practolol, which exhibited cardioselectivity, beta 1 selectivity. And Black was thrilled, and by that time he left ICI and he then went on to discover cimetidine, which I told you about in another company. But after he left, ICI found that even Practolol had problems, and they kept on tinkering. You know? And what they said over here was that Practolol had to be withdrawn because of side effects. So Practolol also had side effects. It was beta 1 selective, sure, but there were side effects as well. And a very, very subtle change with the made here, instead of it an acyl amino group, they just made it a simple amide, terminal amide. You can see CH2CO NH2. This molecule was retained its beta 1 selectivity and it had absolutely no side effects. Okay, and this was that drug which people talked about, which was one of the greatest discoveries of the 20th century. Okay, but what I want you to see was that look, there is a lot of opportunity for, I will say, intuitive reasoning and trial and error, but it's done by subtle manipulations. You keep doing everywhere you can see. Two chlorides replaced by a naphthyl, problem, put in a ether, problem, get rid of the naphthyl, now put in an acyl amino group, still problem, make it into an amide, leave the rest of it intact. Okay, and look, when they were doing all their work, those were early days. Today, if you were to do it, you've got many more tools at your disposal. Okay? Like I'll show you the next advance. You'll see certainly the approach was a little more scientific. Now, look at this molecule, atenolol. Do you see atenolol? Is there any opportunity that you can see to do any further work on atenolol?
We can just have the amine group, that is NH2.
It takes a long time to discover a drug, and once you have discovered, you don't want again a radical change. Can you think of anything else?
Sir,
Yeah.
can we connect the amide thing directly to the benzene ring and eliminate that middle carbon?
Yeah, so you are saying just make directly CONH2.
Yeah.
Without the CH2. Huh?
Yeah.
Again, I'm saying that you'll be going back to discovery research. It might work. Let me tell you, or they may have done it. It may be that this was found to be the best and they could have made all these molecules.
Hydroxylation of alkyl group.
Hydroxylation of?
Other alkyl group.
Other alkyl group. Again, what I'm saying is, for you it may seem like a simple change. Now you can see how simple changes can create havoc. One case, there is it becomes an alpha receptor additionally. Then it affects the blood-brain barrier. Who knows what will happen if I put in an hydroxyl group? Again, I'm going into drug discovery. Now remember, once you have made a drug, the clinical trials, it's a fantastically costly thing. And so again, going back into clinical trial is always a problem. Anything else that you can do? Stare hard at this molecule. I'm asking you for something a little more, I would say, surefire, where you can't go wrong. Brilliant. That's all I wanted. Okay. Okay, so you can see that OH, that's a chiral center. The previous lecture was asymmetric synthesis, and we learned that both chiral forms need not work the same way. All these people, when they had made racemic atenolol. And I was just thinking that James Black or anyone, none of them pointed out about the chiral potential. In those days, you remember 58, 60, asymmetric synthesis had not started. And so people were not really looking at that so seriously. All I want to tell you is that it is the S-Atenolol, okay, which is the active, not the R. And there's a company in Pune which actually was the first to introduce the S-Atenolol, okay, company called MQ. So that is something I find that we in India are quite good at, the process chemistry part of drugs. Fortunately, over here, the other enantiomer is not really very toxic. But between the two, it's only one that is active. Okay, so it's not toxic. So is there any, what's the problem if I use a racemic drug? Higher? And so what is the negative implication of that? You're absolutely right. Very nice answer. Obviously, I'll have a more metabolic load. The liver has to metabolize more drug. Okay, you don't want that. We talked about the beta blocker, and the ultimate one was atenolol, which even today is a very popular drug. Now that many other drugs have come into the market, the general feeling is that the first line beta blockers are not as effective in preventing stroke and total cardiovascular events as first line diuretics. These are things that make you urinate. And so the problematic chemicals get out of the body. And there are drugs for inhibiting what is known as the renin angiotensin system and calcium channel blockers. This is the third approach. What we'll be talking about is the thing that has become one of the really blockbuster approaches and drugs arising from the renin angiotensin system. Just like we saw last time, pioneering work by Alquist and Langley, and how they came up with the receptor. Similarly, here, this was a discovery actually made in Argentina, and what they had found is that when they tied off the kidney, one of the kidneys, what they found is that it leads to a increase in hypertension in dogs. They clamped the renal artery, and later on it was found that it induces the kidneys to form more of a particular compound, which actually leads to high blood pressure. So that was what ultimately led to the origin of the problem and what is known as today the renin angiotensin system. And so sometimes these are quite remarkable, and that work of course was done along with this very famous laboratory called Eli Lilly in Indianapolis in the US. So it was the Argentinian researchers and Eli Lilly who really made this great discovery. Now, let me quickly show you what exactly is this renin angiotensin system. Now, what happens is that there is this particular protein, it's like a polypeptide, you can say, which is produced in the liver, and it is called angiotensinogen, AO. Okay. Now, the stuff which was being produced by the kidney, they found that it secretes more of when they tied off one of the kidneys. That is renin. You have heard of renin. And what renin does, it basically cleaves it. A part of the peptide chain is cleaved off, and it only leaves this part. Okay. And that was named as A1, which is angiotensin 1. Okay. So it was a cleaved product from the original angiotensinogen AO produced in the liver. Now, then what was found is that there is another secretion in the body, an enzyme. It's not a secretion actually, it is tied to cell walls, and that is known as angiotensin converting enzyme. And what it does is that it further cleaves A1 over here and then makes a smaller peptide chain. Okay. And this particular thing was called as A2 or angiotensin 2. This is angiotensin 1, angiotensin 2, and this is the original angiotensinogen produced by the liver. Now, what was subsequently found is that angiotensin 2, A2, is a vasoconstrictor, means it constricts the blood vessels. And because of the constriction of the blood vessels, the pressure goes up. And so that was identified as the cause of the heart problems, the high pressure in many people because of this particular constriction due to angiotensin 2. Now, angiotensin 2 also gets degraded further by another enzyme called ACE2. This is known as ACE only. It's not known as ACE1, but ACE2 cleaves it off here. The aspartate is cleaved off and it forms this. This is harmless. So A3 is not harmful, but the problem is that it is not that the conversion of A2 to A3 is instantaneous. So there is a substantial amount of A2 which is there. So there will be this problem of high pressure. Now, what you can see is that while constructing a drug, you could actually target many things. Like, of course, it is difficult to not have the liver produce this. It's also not so easy to really inhibit the action of renin because the kidneys are producing renin. But what one found is that there was an inhibitor for this particular enzyme, this particular action, and that is known as ACE inhibitor. That was a very popular drug which came up after the beta blockers, the atenolol. But the problem is that the ACE inhibitors give you a side effect, mainly a dry cough. People are constantly coughing, a dry cough, and so people were therefore looking for an alternative to the ACE inhibitors. Now, there were certain polypeptide mimics, which were they kind of fool the receptors on which the angiotensin 2 A2 sits into believing that that is A2, but those polypeptides are benign. So what they do is they dislodge the A2, and therefore the vasoconstriction effect goes down. But what was known was mostly peptide. And in those days, I'm talking about the early 80s, it wasn't so easy to make those kinds of peptides. And typically, in the area of drug synthesis, drugs were mostly small organic molecules. Need not be small, maybe a little more complicated, but they were typically, synthetic organic molecules. Chemists were comfortable with making molecules, but such molecules did not exist as an antagonist for A2. Okay, and then as I mentioned, it is this that happens. Now, this A2 actually can go and sit in two types of receptors, which are known as AT1 and AT2. And it was the AT1 receptor, if it goes and sits, this was the one that was found to actually increase the blood pressure. And so then the target became very clear, just like we saw in the case of James Black, that he targeted the beta 1 receptor. Here, it became very clear that one would need to target the AT1 receptor. And again, this is from the discoverer of the drug. It says that quest for a A2 antagonist was partly on this count, which is the dry cough, and by blocking AT1 receptors, angiotensin receptor blockers, ARBs, lead to lower blood pressure because the A2 is dislodged from the receptor. Okay, so is that clear? I hope everybody has understood this. All right. Now, what is very interesting is that this ACE, which I've talked to you about, one is you can see that it converts angiotensin 1 into 2, and then ACE2 converts 2 into 3. So this is benign and this is harmful. But this ACE2 is not just present in the blood or in those arteries. ACE2 is all pervading. There are all kinds of cells in the body where ACE2 resides. And guess what? The coronavirus is actually attacking ACE2. Okay, this is from Nature. After the mechanism was worked out, that what exactly is the SARS-CoV-2 virus doing, what they found is that it goes and uh, sits on the ACE2 enzyme and then penetrates the ACE2 receptor. All right. And then over here, it obviously gets all the nourishment, from the cell. Okay. And then this part, which is the RNA, gets released. And then once the RNA has been released, it can replicate itself into the protein, and it produces many more of these SARS viruses in the body. And then ultimately it gets out of the ACE2. And that's how the whole problem of SARS begins. And subsequently, of course, there were many defense mechanisms which have been identified. Things which can gobble up the virus, for example, or there are things which help in the gobbling up process, or there are also these cells, B cells and T cells, which actually prevent the virus from binding to some of these problematic receptor sites. And these B cells and T cells have pretty good long-term memory. So they remember, and they're there in the body. Moment, the virus comes, then they get activated, and, that's how you kind of impart immunity to the body. And then, of course, people started looking at also vaccines, which can also similarly trigger an immune response, and that is what has been going on for the last 12 months. Tremendous work in the area of vaccines. So you can see there's a connection between this pandemic and the ACE inhibitor that I mentioned to you about. Now, just like I told you that there are different approaches to solve a problem, and we talked about the beta blocker, and now we are talking about the renin-angiotensin system. All of it is really to prevent cardiovascular disease. Basically, that's the objective. And now you'll also see that, for example, there are so many vaccines which are coming into the market, and they have different ways in which they act. And I'll send you a very nice article which actually tells you what are these platforms, just so that it's such an important area, and it'll be nice for you to know. So there is, for example, one approach which is known as an inactivated virus. That means I make the virus benign, but then it's there in the body, and it kind of helps to compete with the problematic virus. There's also something known as a viral vector. So, for example, the AstraZeneca vaccine that we're talking about, those are based on this kind of viral vector. The Russians as well as AstraZeneca, these are the two who have mostly developed. This is the Sputnik vaccine, and this is AstraZeneca. And the inactivated virus was first developed by China, the Sinovac virus, and in India also they have done it. This is that Bharat Biotech who have worked on inactivated virus. Subsequently, there are also certain subunits of the protein which people have targeted, and this is again another very important vaccine which is emerging called Novavax. And the ones that you're probably hearing about a lot are these two, the Moderna and the BioNTech Pfizer vaccines. These are based on the RNA. I showed you the RNA. This is the RNA. Okay. So they basically go and interfere with the working of the RNA. And that apparently, this is the first time ever that a vaccine has been developed based on RNA. Whereas all of these were known approaches. These are very standard approaches, but this was a completely new approach, which has never been done before. And by the way, I thought that this is important. Many times, when we are in a problem, we always get stuck with a single solution. You always think about that, how to improve it, how to do this. But to think out of the box and to look at another completely different solution, that is the kind of thing what innovation is all about, and what extraordinary research is all about. It's no different when I was thinking that you look at a fixing of atmospheric nitrogen within 10 years in the first from 1900 to 1910. Look what happened. We started with the Birkelandite, which was the electric arc process, and how it was nicely stretched out into a disk with the help of a magnetic field, and 1,000 times increase in throughput. Extraordinary. And then you had the cyanamide, the calcium cyanamide approach, completely different. It's got nothing to do with the Birkelandite electric furnace, electric arc process, although in this particular case also it was a high temperature process. And then you have this remarkable work by Haber, and coming up with the synthesis from H2 and N2. Three totally different approaches within 10 years. And look at the number. 56 COVID vaccines are today in the clinical trials. There are another 166 which are in preclinical stage. What does it mean? When there is a sense of urgency, your life depends on it. You will be doomed if you cannot solve the problem. That's when people really wake up, and you have this unbelievable spurt of activities unheard of, that so many solutions were found within 12 months. It's incredible. you know so many solutions to such a difficult task like fixing of nitrogen found within 10 years. After that, I have not seen in the next 120 years that kind of remarkable breakthroughs. It's simply because people have not been working on problems which are really life and death issues. They're working on nice to do things. Nice to do things are things which you may do, may not do. But when your life depends on it, then you'll do this. Your life depends on being able to generate urea, generate nitrogenous fertilizer. It's not a joke. I mean, otherwise people will all die. And people realized that, and they had that sense of passion and sincerity. And so I hope you can see. So this was just a digression from what I want to talk to you about. Okay. So coming back to this now, all right, I will tell you the journey that was followed in making an antagonist for A2, the angiotensin 2. Now, I am the most fortunate and luckiest and privileged person, okay, because the discoverer of this happens to be John Vititus Dancia. We were the closest of friends from day one when we both entered for our PhD over there. He was hired by DuPont, and he worked on this particular problem. And John was awarded the Lavoisier Medal by DuPont. And if you read some of the names and what they have done, you will know how great an award this is, because we are so close even to this date. So I could have a constant dialogue with him. He sent me all the material of the entire discovery process so that you'll know exactly what he and his colleagues did.